Good morning.
Good morning, sir.
Good morning, sir.
Good morning, sir.
Yes. Okay. So today also we have 18 students on board. Fine. So let's begin. So since last 2-3 days, we had been discussing about the metabolic network, that how to go for the generation of a network, the metabolic network. So uh, as for that, as I told you, that we should know the system. So we have seen many aspects whereby we try to understand that what is actually happening into the cell, that there is something which is coming inside, something which is going outside. Okay, and within the particular cell there are many metabolic reactions which are going on, and all these metabolic reactions are interconnected to each other, right? So if you want to go for any kind of modification, first we'll have to propose your own model. Okay, and for that generation of that model you should have the different type of the networks. Okay, so we have seen that there you can have a metabolic network, you can have a regulatory network, you can have a signaling network. So and like this. So but these are the three which are majorly use type of the networks which we'll have to build up first. And then the topic what we saw yesterday was about the systems biology. So for the development of all these different type of the networks, you will require to study the cell as a whole. So you'll have to study it with all the respects. Means what kind? Means you should have the genetic information. Means you should have the genome information, the transcriptome information, the proteomic information, then the metabolomic information, then the flux information, or if there is any kind of interaction between the biomolecules, so like DNA protein interaction, protein protein interaction. So all these things should be studied. Then only you can develop your own network. Fine. So both these things which we have talked about, but today we'll go something in detail about the regulatory network, because that is one of the aspect which is very important. Within the cell, what we have seen, that the cell will never go on producing the energy continuously. It will never happen that the cell is producing only one of the product, which is that one of the product is continuously produced. Okay, or that is what means the energy is not continuously produced, or the products are not continuously produced. There is a very good amount of regulation, and this regulation is going to help us to understand that what kind of modification will bring about a change from genome to the phenotype. Okay, so the production of a particular metabolite, as we know, it is going to reflect the phenotype of that organism. So if you have a proper regulation of that particular pathway, whereby you are trying to express that particular metabolite at a very maybe at a higher concentration, or you are trying to suppress the expression of that particular metabolite, obviously it is going to affect the phenotype, right? So in that sense, today we may talk some details about the regulatory network, that why it is so important. Okay, or you might have seen that if the nutrient is changed, okay, if the nutrient is changed, the cell may behave differently. Okay, so a few of the examples which I will bring about, which you might have already studied during your molecular biology lectures, but it is very important that you should understand this regulatory network somewhat in details. Okay, so to continue, I will be presenting the screen, and then we'll try to see today about the regulatory network. Okay, so and then after understanding this regulatory network, we'll go for the construction of the metabolic network actually. Okay. Yeah, is the screen visible?
Yes, sir.
Okay. Fine. So this is what the regulatory networks what we are going to talk about. So this also, which I have told you, that what do you mean by 1D annotation? 1D annotation is simply that you have the genomic information, and from that genomic information you can segregate them in the form of ORFs, that is the open reading frames. Okay, and these open reading frames, what we have identified, we have assigned them. Okay, we have found that these particular genes or the ORF, they are responsible for one particular product. So according to that, one example which I have quoted, that in case of E. coli, you have around 124 or 126 different type of the classes of these ORFs which can be generated, and which are related to the maybe to the transporters, maybe to some specific pathways, or some particular metabolic metabolism of carbohydrate or proteins or nucleic acids. So or some of them will be for the regulatory mechanism, some of them could be for your signaling mechanism. And from that one dimensional annotation you can go for the two-dimensional annotation. Means what? Means we are trying to study it as a whole. Means you have the ORFs, you know the genes, now you know the products of that particular genes. There is someone who wants to enter. uh, yeah, so you have the genes. Okay, you know the gene products. Now the gene products are what? They are nothing but the proteins, and these proteins are going to carry out the different kind of functions. So there I told you that it could be a signaling function, it could be a regulatory function, it could be the catalytic function. Okay, and for having these different type of functions, again these gene products can interact with each other. Okay, so you can have your matrix in which you are trying to explain whether there is an interaction between the different genes, whether there is an interaction between the gene and its gene product, or whether there is an interaction between the gene product type of an interaction is there. Okay, and based on that you can generate your own matrix which will help you to develop the complete metabolic network. Okay, so that is what I have explained you in the last lecture about the from the individual component to an two-dimensional annotation, right? So to continue with that, there are two types of networks which can be generated. So you can see here a comparative account which is given. One can be called as what? The biochemical reaction network, and other can be called as the statistical inference network, right? Now the basic difference between these two is that the biochemical reaction networks is a realistic. Okay, I'm talking it that it is a realistic. Means what? You are actually studying the system, you are actually studying the biochemistry, the genetics, and the various aspect of that particular cell, that what actually is happening into the cell, and from that particular information you are trying to develop a network. So that is why we can call it as a biochemical reaction network, right? So you can see here one picture which is given, that you first try to understand that if A has to be converted into B and B has to be converted into C, so for that you require the enzymes. So you are studying all those particular details which are actually happening into the cell, and then you try to develop a network. So that kind of network is called as what? The biochemical reaction networks, right? On the other hand, you have another type of network which can also be generated, which is the statistical inference networks. Means what? You have too much of data which is available. So the data could be your genomics data, it could be the transcriptomics data, the proteomics data, and from that particular data, with a notion, what we know that the genomic data can be correlated with the transcriptomic data can be correlated with the proteomic data, and from the proteomic data you can correlate the metabolomic data, right? So though the data which is now available, which we can obtain from the different type of the advanced instrumentations what we have, so that data we are trying to mathematically or statistically put in a form of an network. Okay, and that is nothing but the statistical interference network. Okay, so in that you can put all kind of information. So it could be your the actual what you can say, the component data, that is genome, RNA, that is DNA, RNA, protein, or the metabolite, and as well as you can also put upon the type of interactions which could be there between these components, right? So simply remember this. So biochemical reaction data is a realistic, and this is the statistical type of the information which could be obtained from the data which is being obtained from the different type of the omics technology, right? So in the next slide, I'll give you the actual what you can say, the comparison between them. So from this you can now understand when I'm talking about the biochemical. So there you can see here, which I told you, that you require a significant knowledge of the system. You should understand the biochemistry. You should understand that what kind of physics or chemistry which can be directly applied there, because all these physics chemistry laws are applied to our living system. Then you can relate more closely to the phenotype, means with respect to the change in the fluxes which I told you. Fluxes of what? The metabolite. So if there is a change into the concentration of the metabolite, it is going to affect the phenotype. Okay, so this network what we are trying to develop can be closely related to the phenotype of that particular organ. Now once you have developed a biochemical network, you cannot change it. Why? Because it is a fixed, which if you remember, I told you that it is a blueprint. The interior of the cell, whatever is happening into the interior of the cell, it is not going to change. It is going to remain as it is. Whatever may be your input, what you have put in, right? And that is why this biochemical network can never be changed. Okay. On the other hand, if you look at this statistical network which you see here, okay, so I'm not going to explain you this, that how this network is developed, but we are just trying to see that how these networks are developed and what kind of information you can get. Now, for this development of this statistical network, you do not require an complete information of the system. There is no need of knowing the biochemistry. There is no need of trying to understand that what physical and chemical laws are going to affect, etc., etc. Simply what we are doing, we are taking the available database which we have obtained from the genomics, proteomics, metabolomics, or the transcriptomics studies. We put it into a form of an program and try to develop a network. Okay, so you can see the fourth point which I have written here. So you can relate more directly to the high throughput data what you are getting. Okay, so from that particular information, we have done some statistical calculation. We have put down it in the form of an matrices. We have tried to see whether there will be an interaction or not, etc., etc., and we have tried to develop an network. Now in the statistical form, when you do it into in silico, there you can easily modify or reconstruct the network. Okay, so the network wiring can be changed in case of the statistical network system, right? So just remember that these are the two types of the network models which can be generated. So the actual point what we are trying to understand is about the transcriptional regulation. It's not actual transcription, but what I want to say, the regulation, why it is very important, why it is important. And within that you have a type of regulation which is called as a transcriptional regulation. So if you remember in the yesterday's lecture, I showed you one diagram that if you are converting a substrate into a product, okay, so that product which is formed will be having two types of control. One is the metabolic control, means the allosteric mechanism, what we know, that the end product is going to inhibit the first enzyme of that particular pathway. Okay, so that you can call it as a metabolic regulation. And the same product what you are getting, it can also regulate the pathway at the transcription level, okay, at the expression level you can say. So there also you can say that this end products, it can have the transcriptional regulation, right? So what is this transcriptional regulation? If you have understood the different type of the regulations, this transcriptional regulation, they can be imagined as the on and off switch at the genetic level. So whenever required, the genes are expressed, and when not required, the genes are suppressed. Okay, this present situation, that you people are simply listening, you are not doing any kind of work. So maybe in your case the enzyme levels are constant, but in my case I am speaking, means I am doing a good amount of work here, so my energy requirement is very low. Okay, so a particular X gene in your case is constant, but that gene in my case it is expressed at a very high rate because I am requiring the energy at very high amount. Fine. So in your case you can say that the genes are now off, while in my case the genes are now on. Okay, so if I stop talking, if I sit like you, that I am simply listening, so in that case what will happen now, that my genes, those particular genes will be now off. Okay, so that way you can imagine that this transcriptional regulatory networks, they are the on and off switches. So that is what I am trying to explain here. So obviously for any kind of gene expression, you can say, or any kind of pathway to be activated, you require a signal. So you have a specific signal. So this signal is going to activate something called as a regulating component. So it could be any kind of protein, what we have seen. So specifically we have seen that they are the kinases. So you have those particular regulating component which are uh, activated. Now this regulating component will then go and have its effect. Now the effect could be at what? It could be at the DNA level. Okay, so that is, it will not allow the transcription to take place, or it may not allow the translation to take place. So obviously it is going to have the effect onto the amount of RNA or the amount of protein which is being produced. Fine. So here it could be either overexpression or it could be suppression. I'm not talking about any one of these. So when I'm saying a signal can either activate or it can suppress a particular gene. Okay, so the signal is activating a regulating component. This regulating component, now in this case we are talking about the suppression. So this regulating component, it is going to change the RNA or the protein output. And if there is a change into this RNA or a protein output, what is the endpoint? What you get? That you get the change cell behavior and the different type of the structures which can be generated. Okay, so whatever activity, for whatever kind of activity where this regulating component is affecting the gene expression, that activity will be stopped. Okay, means if you have taken a food which is very rich in carbohydrate, so in that case you can say that the genes of the glycolysis enzymes, the gene expression will be very high. The moment the glucose level has dropped down, so this gene expression will be suppressed. Right. So it is the input signal which is affecting the behavior of the cell. Okay, and that is through this regulating component which can bring about a change into the RNA and the protein output. Right. So what you can say, that the input signal is going to affect the phenotype of that particular organism. Now all these entities which I have mentioned, these four blocks which I have mentioned, they are related to each other, where they can have interaction within themselves. So the input signal can directly affect the phenotype change, it can affect the regulating component can affect the RNA and protein output, and so and so. So they are interrelated or interconnected to each other. Fine. Or you can say that each one of them, they are dependent on each other. Right. Now, on the other hand, the output what you are getting, now this output is going to affect the input signal. Okay, see, if you're making a change into the genotype, it is affecting the phenotype. So that phenotypic change which has taken place is again going to regulate or affect the input signal, right? So this loop is now completed. So you can see that this output what you are getting, it is going to affect the input signal, so that the input signal will be removed now. Like if the glucose concentration is very high, you can see that those genes are highly expressed. But the moment the glucose concentration has lowered down, the gene expression will be suppressed. Okay, So what is going to happen here? Why they are going to suppress? Because the insulin level is going to drop down. So something like that you can now imagine here, right? So this is how the transcription regulation it plays an important role in the overall functioning of the cell. Okay, and that is why I said that you have to give a more stress onto this regulatory network. Now, why do we care about the regulation so much? Okay, why I'm emphasizing more onto this regulation mechanism? Because this regulation, it has a significant effect on the cell behavior. Just now I have explained you that depending upon the need, either the genes are expressed or they are suppressed. Okay, so always these pathways or the different type of the signals which are taking place within the cell, they are under the control, and that is why you can have a stable system which can be generated. Okay, and this has been proved by doing some experiments. So in case of E. coli, what they have studied, so they have found that there are something 400 regulatory genes. Okay, and out of these 400, 178 regulatory and are the putative regulatory genes found into the genome. Putative manje kai means they are continuously responsible for regulating the different type of the pathways. Okay, so there are 690 transcription units which are there. Okay, and these transcription unit are under the regulation of this 178 regulatory genes. Okay, there is one database which is called as a Regulon database. So if you visit the website of this Regulon database, you will get all the information about the regulatory net, right? So at the end, you can say that this regulation will have a major effect on the model predictions of the cellular behavior. Okay, so that is why it is very important. Now you might have studied about these different type of the regulation systems. So the operons are the very best example here. So you have the lac operon, you have the tryptophan operon, you have the galactose operon. Maybe I'll not explain you all this in detail. I think so in molecular biology, you might have studied about the operons. Here, what I want to emphasize that you have the carbon source. So if you vary this carbon source, there will be an variation into the operation status of that organism. Okay, so this is what only the comparison which I wanted to show you. जे आता मी तुम्हाला सांगितलं that the carbon source is going to affect the operational status of that particular organism. Right? So what happens in lac operon? That instead of using an easy source of carbon carbon, that is the glucose, what we are doing, we are adding the lactose into the medium. But the E. coli system, it cannot easily what you can say, use this lactose. Okay, so but if you are allowing this organism to grow into a medium containing a lactose, so in that particular condition, what we see that there are some specific set of genes which are expressed in presence of lactose. So those genes, when they are expressed, at that time only the lactose can be utilized. Okay, and once the lactose has been finished from to the medium, in that case what will happen, again this gene expression will be stopped. Okay, so it's a like of an on and off type of a switch. Okay, so whether means the moment the lactose is there, the gene expression is on, and the moment the lactose is not there, the gene expression will be stopped, right? So the lac operon you can now understand here. So look at the end point. So in presence of glucose, the expression is off. So in presence of lactose only, the expression is on. Okay, because it is affecting, it is binding either onto the promoter site or the operator site, or there are some repressors which are generated, etc, etc. So only in presence of lactose, the RNA polymerase can bind to the what you can say that cascade, the ORF for these particular genes, and the gene expression will take place. So if neither glucose or lactose is present, so in that case also you can say it is off, or if both of them they are present, so in that case also it will be remaining to the off position. Right? So it has to be only the lactose which has to be present into the medium, then only these genes are expressed and the E. coli can grow into that medium. The same way you have the galactose regulatory system, you have the tryptophan regulatory system. Specifically, I have just put on these two things only. So if the medium is not containing glucose as well as galactose, so in that case, these genes for the galactose regular metabolism, they are off. But if it is only galactose which is present, so in that case, the RNA polymerase will bind and the mRNA will be generated. So the genes are on now. Okay, and if both of them they are present, so in that case also the RNA polymerase cannot bind and your gene expression is off, right? So I think with these two examples of the operons, you might now aware that why this this regulatory network is very important. So there are three fundamental data types which can be thought of when you're talking about the regulatory network. So in all these three data types, we are trying to work at the different levels. So the first one is the component data. Okay, means what actually is being inhibited so that the regulation is taking place. So in this case, the data types, which it could include the components like the binding sites. So it could be your polymerase binding site. It could be the transcription factor molecules. Okay, means whether the transcription factor molecules they are active or inactive. Okay, so that kind of data. Then you have something called as an ribo switches, and so and so. Okay, so all those entities which are required for the actual transcription of a particular gene. So if you have a data of all those things, that constitutes the component data because they are the component for your gene expression. Then you have the interaction data. Interaction आता तुमच्या लक्षात आलं असेल that it has to be an what you can say binding or moving apart from each other. So there is a chemical interaction which could be there, and here the interaction which it could include the DNA protein interaction, it could include the protein protein interaction, it could include the metabolite RNA interaction, etc., etc. Right. So if you have a data of all these kind of interactions which are taking place when the regulation mechanism is on, so that kind of data is nothing but the interaction data. Okay, and at the end you have the network state data. Okay, means a fixed state, means if the glucose is present, okay, so that whatever the data you have with respect to the genome or the transcriptome in that particular condition. Okay, or if galactose is present, what is the state of the DNA, RNA, and the protein in that particular state? So that kind of information is nothing but the network state data. Okay, so the state of a whole network can be assessed by a variety of data generated from living cells in well-defined environments, such as genome scale expression data and the phenotyping data. Right. So these are the three fundamental data types for regulatory networks, which you have to take into consideration. The bottom-up data types, again, these two, what you can say, means from those three, what I have explained you can have these two types of the data types. So bottom-up data types, what is this? So this is the data which is derived from classical biochemistry or genetics that are focused on a single or a few variables are often referred to as bottom-up data. Okay, means again the example of lac operon. So you have the condition where glucose is present, you have a condition where lactose is present. So you have these two different states of the organism. Okay, so you have the component data, you have the interaction data, you have the network data. Okay, so all those three type of the data, if you have, so that is called as what? The bottom-up data. Okay, so if you visit the Regulon database, which I told you, within that database you may come across the different databases which are available. So you may have one of the site having the E. coli regulation database, someone having the yeast regulation database, the human regulation database, etc., etc. So like this, there are many different type of databases which are available. So that may constitute the bottom-up data types. Okay, So the construction of these bottom-up data types is laborious because here you'll have to study the component by component. तुम्हाला एक एक पार्ट अँड पार्ट तुम्हाला त्याच्यातला स्टडी करावं लागेल. ते स्टडी केल्यावरती जे तुम्हाला इन्फॉर्मेशन मिळेल त्याच्यावरून तुम्ही तुमचा डेटा सेट तयार करणार आहात आणि मग त्याच्यावरून तुम्ही नेटवर्क करणार आहात. So in that sense, these bottom-up data types, they are very laborious type of the data types. Okay. But nowadays, as I told you, you have the ready-made websites whereby all these databases are easily available. So simply you have to take the system in which you are interested, take the information and try to go for the work. Okay. So you have the bottom-up data types, and the other type is what? The top-down data types. Now this top-down data types is similar to that of the neural network, the statistical network which I have explained you, that we rely on the high throughput data which is being obtained. Okay, so what is done here, that you take an E. coli growing in glucose. Okay, so in that particular condition, you try to have all the genomic, proteomic, transcriptomic, metabolomic information. Okay, and then you develop the network. Take the same E. coli, but now you put it into lactose medium. So in that particular state, again you go for the genomics, proteomics, transcriptomics, metabolomics studies, get the data, and then you put it into a form of a network, right? So such kind of data which you have to construct the network is called as what? Top-down data types. So it can be done in three ways. So the first one is what? That you can experimentally determine the expression state of a genome. Okay, so you, as I told you, that you can have your organism in the different state, and then you go for the complete data. Go for the complete data sets. The complete data set म्हणजे मला इथं म्हणायचं की तुमचा complete genome profile, त्याचा RNA profile, त्याचं protein profile, त्याचं metabolite profile. हे सगळे तुम्ही त्या कंडिशन मध्ये घ्या. परत कंडिशन चेंज करा, get all the information. Again change the condition, get all the information. Right? So in that condition, you may come to know that some of the genes are overexpressed, some of the genes are suppressed. Okay, so such kind of variation you may get it, and now this variation you can now try to prove it. So there is a thing which is called as a knockout. I think all of you know what is knockout. Okay, or maybe when we study the genetic engineering in the next semester, at that time we may come across this term called as a knockout. Knockout मध्ये काय करतो, we do not allow that gene to function. Okay, or that particular gene is being. मग जर आता ह्या एक्सपेरिमेंटल स्टडी मध्ये आपल्याला समजलं की एखाद्या जीनचं एक्सप्रेशन होत नाहीये किंवा एखाद्या जीनचं एक्सप्रेशन खूप जास्त होतं, आणि मग ते जर आपल्याला प्रूव्ह करायचं असेल, सो व्हॉट वी डू, वी ट्राय टू क्रिएट दिस जीन नॉकआउट्स. Okay, and allow it to grow, and then you have the experimental data sets which could be up, and then you compare with the above data, and then you can say whether the things are correct or not, right? So the first one, as I told you, it is experimentally determining the expression state of a genome. The second could be identifying all the promoter sites using computational approaches. Means what you can do, you have the complete genomic data, you have the complete transcriptomic data. Okay, now you have this data which is available. So what you can do, you can go for some computational methods. We try to find the homology between the different type of the transcription regions. So in that case, what you may find, that there are some hundreds of genes which are having the same kind of the transcription factor. You may find some 50 genes which are having somewhat different type of the transcription factors. So from this, what we can correlate, that those genes which are having the same kind of transcription factor, they might be having the same type of the gene expression profile. Okay, म्हणजे आता आपण हे बघा, पहिल्या याच्यात काय केलं होतं, आपण पहिल्या याच्यामध्ये वेगवेगळ्या स्टेटला त्या जीनचं एक्सप्रेशन होतंय नाही होते ते आपण बघितलं. ठीक आहे, ते आपल्याला काय माहिती मिळाली. आता देर आर thousands and thousands of genes, सगळे एकत्र आपल्याला स्टडी करता येणार. सो वॉट एव्हर द इन्फॉर्मेशन यू हॅव गॉट, फ्रॉम दॅट वी आर सेइंग that this particular transcription factor is affecting the expression of the gene. आता एका जीन बद्दल आपल्याला ती माहिती मिळाली, पण असे thousands of जीन आहेत, मग सगळे कसे स्टडी करता येतील आपल्याला. सो व्हॉट यू कॅन डू, इन कॉम्प्युटेशनल मेथड्स किंवा इन सिलिको मेथड्सने आपण काय केलेलं आहे की हे सगळे जे जीन रिजन्स आहेत ते आपण कम्पेअर केले, त्यांच्यातली होमोलॉगी शोधून काढली, जे सारखे आहेत त्यांना बाजूला केले. ओके, आता सपोज ज्या जीनची माहिती मला आहे की विथ रिस्पेक्ट टू इट्स ट्रान्सक्रिप्शन फॅक्टर, सो इफ आय गेट अराउंड सम मोर hundreds of genes which are having the same kind of transcription factor, so I can say that the expression profile for these genes is also similar to that of the one which I have studied. Fine, so that is the another step which we can do, and at the end you can experimentally determine the location of protein binding sites on the DNA. Okay, so there are different methods which can be used. You might have heard about chromatin immunoprecipitation, that is called a chip. So you can go for the different microarrays. Okay, so that is the chip what it is called as, or nowadays what we have massively parallel sequencing, that is chip sequencing. Okay, so these are the different methods which we can study, which we can do, and from these experiments we can find out the protein DNA type of an interactions. Okay, and if you know these protein DNA interactions, you can easily correlate it with the transcription of that particular gene. Okay, so this is how the top down data types are generated. Okay, but now there are many issues with this top down reconstruction. What are those constructions? That these are very complex models. Okay, and the algorithms which are generated, again they are complex. Now why is that they are complex? See, we are relying on what? The high throughput data. Okay, so to have this top down reconstruction, we are relying on what? The high throughput data, and for this high throughput data we are using some specific techniques. So it could be your sequencing techniques like ट्रान्सक्रिप्टोम सिक्वेन्सिंग असेल, जिनोमिक सिक्वेन्सिंग असेल, किंवा तुमचं प्रोटीन प्रोफाइलिंग असेल, किंवा मेटाबोलाइट प्रोफाइलिंग. सो वी आर डिपेंडिंग ऑन दिस हाय थ्रूपुट डेटा. But now we are not sure that whatever the high throughput data what you are getting, whether you are getting the complete information, whether you are getting the correct information. Okay, see, you get the data, you get the information, but you are not sure that whether you have got the complete or the correct information. Okay, so that becomes one of the disadvantage of this top down reconstruction, right? So currently whatever the information or whatever the knowledge what we have, it is a type of an hypothesis which we have generated. Okay, and we are still working on that particular hypothesis. Still we are trying to get some accurate type of an information so that we can develop an actual network for the regulation of these different metabolic pathways. Okay, so whatever the regulatory networks which are available, it could be a combination of your bottom type databases or the top down databases. Okay, so we are taking the information from all the possible databases which are available. So you can see here that based on our knowledge and based upon the data what you get, so having the information from both these things, that is knowledge and data, you can develop your regulatory network. So what kind of knowledge you should have? That is the genome annotation nowadays which is available. There are many systems whose genome is now available, and if your genome is available, you can have that genome which is annotated. So you have that kind of knowledge. You have the literature मध्ये तुमच्याकडे books आहेत, there are many review articles, research articles are there. So you have that kind of literature, so that knowledge is available. You have the different databases nowadays which are available. So all these things, they what you can say, constitute the knowledge what you can get. And on the other hand, you can work upon the system in which you are interested. Then you can go for your actual database for that system. So it could be your promoter sequence, the gene expression sequence, data sets, or the location analysis, the different type of the genomics, proteomics, transcriptomics, metabolomics सगळ्या प्रकारचा डेटा तुम्ही मिळवा. So your data and the knowledge which is available, you combine, okay, and you can get this regulatory network which is obtained, right? So this is how you can go for the regular network system. And at the end, what I can tell you, that this regulation is at different level. Okay, so there are different factors which are affecting the regulation of the metabolic pathways. Okay, so you can see here that suppose there are in E. coli, it has been worked that there are around 479 metabolic genes which are available. Now these 479 metabolic genes, they are regulated by 104 transcription factors which are generated. Okay, so you can see the complexity, that there are many transcription factors which are affecting the expression of these metabolic genes. On the other hand, these 104 transcription factors, okay, so they are stimulated by around 102 stimuli. So there are different type of the stimuli which are responsible for the generation of the different type of the transcription factors, and these different transcription factors are regulating the metabolic genes. Okay, and these metabolic genes are then affecting the actual metabolite production, right? And for this particular reason, what I have started with, that we should have a very good knowledge about the regulation. Okay, so we should be able to have this regulation network in front of us, in addition to the other things what we have already studied, so that you can go and for an successful uh, metabolic engineering. Okay, so I think with this I have given you an idea about the regulatory mechanism and the network, why it is important. Any questions, any doubts? Hey, you people have already studied these things. Okay, there could be few terms which are new to you, but majority of the basic things you people have learned. Simply what you are supposed to do, you have to apply your logic and try to bring it together, so that you can understand this overall process. Right, so that was all about the regulatory network. So the time is 10:50. Yes, any questions, any doubts, what you have? काय विचारायचंय? I'm not sure whether I should start with the next lecture. कारण की जर सुरू केलं तर आपण कंप्लीट नाही करू शकणार. So it is with respect to the. ठीक आहे. So maybe tomorrow we'll continue. I'll stop here today. So tomorrow we'll try to understand the actual construction of the metabolic networks. Okay, that how will you construct the metabolic network. मग त्याच्यामध्ये तुम्ही रेग्युलेशन नेटवर्क पण इन्क्लूड करू शकतात, सिग्नलिंग नेटवर्क इन्क्लूड करू शकतात, आणि एकदा तुमच्याकडे नेटवर्क तयार झाला की मग त्याच्यावरून तुम्ही तुमचा मॅट्रिक्स तयार करू शकतात, अल्गोरिदम तयार करू शकता, अँड देन computational methods ne tumhi you can study whether the modification what you are trying to do will be and fruitful or not. Okay, and once that is done, you can go for the actual lab studies. Okay, so अरे संपत आला असतं पण ठीक आहे. I'll stop here today. आज वेळ आहे तुम्हाला नंतर कधी दुपारी?
सर मॅम 2 लेक्चर घेणार आहेत, मग टायमिंग माहिती नाही कधी.
ठीक आहे, मग जाऊ दे, आपण आज नको करायला. उद्या मला तरी वाटतंय कोणाचे लेक्चर नसतील. सो टुमॉरो विल कंटिन्यू. सो टुमॉरो मे बी 1-1.5 तास किंवा 2 तास मी घेईल तुमचं. फक्त एक थोडंसं त्याच्यात मॉडिफिकेशन करेल. आयदर ॲट 10 or 11 आय विल स्टार्ट. आय लेट यू नो. आय पुट इट ऑन टू द ग्रुप. क्लासरूम मध्ये मी टाकेल. सो आयदर ॲट 10 o'clock or 11 o'clock विल मीट अगेन टुमॉरो ऑन संडे.
ओके सर.
आणि त्यात थोडंसं अजून एक हे करून उद्या काही पार्ट आपण संपवून घेऊ. so that we'll be in a good shape with respect to the syllabus which has to be covered. I hope you people are getting what I'm trying to explain. थोडस नवीन असतील तुम्हाला काही गोष्टी किंवा एका गो मध्ये कितपत तुम्हाला समजणार आहे ते मलाही आता सांगता येणार नाही, कारण की सगळं व्हर्च्युअलीच चालू आहे. But still I am trying my best through the presentation. I am trying to explain you what actually is required to understand. I have shared you with some research articles and one book, but I can understand it's not a time where you can read the whole book and try to get the information. But if possible, you can go through that book, okay? Or maybe once I'm finished with these lectures, I will share you these presentations with you, okay, so that you can go through these presentations, and you can study only those particular points which I have emphasized. So that can give you an wholesome idea about this topic of metabolomics and metabolic engineering. ठीक आहे, एक दोन दिवसांनी मी तुम्हाला शेअर करतो ग्रुपवरती, कारण की मला ते थोडंफार मॉडिफिकेशन्स करावे लागणार आहे, बिकॉज आय हॅव हाईड सम पॉइंट्स. आय हॅव ते काय म्हणतात, ॲनिमेशन करताना बऱ्याच गोष्टी केलेल्या असतात, तर आय हॅव टू रिमूव्ह दॅट अँड देन आय कॅन प्रोसीड, राईट. सो मे बी